Day3. My name's Holden Maecker, and I'm director of the Human Immune Monitoring Center here at Stanford, and I'll be kind of your host for today. The idea of today is that we're going to introduce you to a broader range of technologies for immune monitoring, since the first 2 days, which featured hands-on lab work, were largely directed at CyTOF and phosphoflow. And so we're going to mention those here and there today, but we're going to largely focus on other types of technologies that are useful. And I'm going to give you a little bit of an introduction to the HIMC, not so much as a way of advertising what we're doing, but to point out what I think are some of the key technologies and strategies that we've used to try to do human systems immunology. So, and then I thought this would work. Never assume that it will work. I think I'm frozen here. Do I need to restart again? Yeah, it went black again when I went to full screen display. It's very weird. If we can't get it, I can transfer to a USB. But at least we have to see something there. Okay. Yeah, thanks. There's this one too. So, do you want to just try again? Let's try. No, it shouldn't. Okay, there's my arrow. And full screen. Voila, okay. So why human systems immunology? Well, much of this has been articulated particularly by Mark Davis, and so you may have heard these kinds of arguments before, but just to sort of quickly summarize, we know that in immunology there's a lot of gaps between what mouse models have taught us and what has worked well in terms of human therapies. And we also know that, and are appreciating more and more, that the immune system is really a complex and self-balancing kind of system. So reductionist approach that focuses on one gene or one protein is not necessarily going to tell you what happens when the complete system is intact. And furthermore, we now have technologies that allow us to collect much more comprehensive data than ever before, and to do so actually in people. And so there's kind of no reason not to use a broader systems approach. But it's not necessarily easy. So things that make human systems immunology difficult is the ability to collect comprehensive data at many levels. So even though the technologies are out there, many labs don't have all of these technologies available to them at all of these different levels. And the whole process of getting clinical samples is not easy, right? It's easier to set up some mouse cages, and even there the bureaucracy has gotten harder. But when you talk about doing a clinical study, there's a lot of expertise that's needed in terms of IRBs, but also coordination and sample handling, how to bank and freeze down samples and things like that. And then the analysis, of course, is sort of the big looming challenge. Once you've done all of this, how do you actually put together all of the data and make sense of it? And so those are things that have kind of impeded the growth of systems immunology in general and in humans in particular. And so that was kind of the impetus for Mark Davis some 7 years ago now starting this Human Immune Monitoring Center to essentially make translational human immunology more accessible to investigators. And so this slide kind of outlines the three goals that we have. First and foremost, we want to be a kind of one-stop shop for immune monitoring. We will meet with investigators and help them with everything from trial design to how to process and bank the samples, which we can do. We perform the assays on various different platforms, and then also do analysis and help with interpretation of the data, which then hopefully feeds back to help design the next study. In addition, we're also a kind of proving ground for new technologies, so having this sort of fertile stream of clinical samples coming through makes it a good place to test out new things and compare them, see how appropriate they are for immune monitoring. And finally, we, unlike the typical kind of core lab, which hands you your data, says see you later, we want to actually hang on to and integrate the data or aggregate the data across multiple projects and thereby provide a kind of repository where we can mine for metrics of immune health versus disease. And as I'll show you in just a moment, that's really only possible by doing a limited set of standardized assays so that you can actually compare the data across projects. And so we've designed a menu of standard assays. Also because if we did not have a limited set of assays that we've standardized, we would never be able to have a throughput that would approach anywhere near what we wanted, right? We would spend all our time on assay design for each specific project. And so instead, we've tried to create as comprehensive of assays as we can in these three general areas, basically cells, proteins, and genes. And we do the cell analysis now by both conventional flow cytometry and CyTOF, and we have these three kinds of assays in those areas. We do the immunoassays primarily by Luminex, and actually I didn't update this slide. We're just about ready to launch a 63-plex Luminex assay for the human and a 36-plex actually in the mouse. So these things have been in flux just recently. On the genomic side, we've done standard microarrays at whole genome and more focused areas. But maybe more interestingly, we have started to develop qPCR arrays on a microfluidic platform, the Fluidigm platform, which I'll talk about a little bit, and also now starting to add to that T-cell receptor repertoires. So I'll take you through a few key assays among this list. I'm not going to go through each and every variation, each and every standard assay that we have, but rather point out a few highlights, which again I think are sort of fruitful ones, critical ones for doing the human systems immunology. And this one, of course, I don't need to introduce to you because you spent most of the last 2 days doing phosphoflow. The particular variant of this assay that we use on the LSR2, on the fluorescence flow cytometry platform, is essentially an 8-color assay. We use 2 colors for barcoding, and so we have a total of 8 stimuli that are different cytokines, and we separate those out by the 2 barcoding colors. And then we have cell surface stains for just 4 or 5 major lineages, B cells, CD4 and CD8 T cells, monocytes, and by negative exclusion we can gate on NK cells fairly well. So that's 4 or 5 different cell subsets and 8 different cytokine stimuli. And then we read out 3 different stat proteins, one of which is shown here. And so you see a kind of typical response pattern for B cells where a couple of these cytokines like interferon alpha and gamma are stimulating strong B cell responses, the other cytokines not so much. And where this has been useful is, among other things, an aging study in Mark Davis's lab led by Shai Shanor, in which the key finding was that young versus old individuals often had a suppression of these cytokine responses that was discernible over a wide range of titrations of cytokine, and in which the older people basically had a flattened response. So higher baselines in many cases and lower fold change in response to stimulation. Not for every single response, but oftentimes patterns of multiple phosphostat responses. And so Shai calls this a cytokine non-responder phenotype. So that's one interesting assay, I think, which we've now also carried over into the CyTOF platform. And so again, I don't have to introduce this because you've been working with it for the last 2 days. But we have 2 of these CyTOFs in the HIMC. We're using them to do anywhere from 30 to 40 parameter assays now, mostly focused on PBMCs, with the obvious advantage that we get much better resolution between the channels and ability to do more parameters in a single experiment. So this is a different kind of assay from PhosphoFlow, but it's one of those 3 standardized assays I showed you that we use, and this is the intracellular cytokine assay, which we've also standardized for the CyTOF platform. This particular example using CMV stimulation. And I'm just showing you some selected dot plots here because there's more markers and way more combinations than we could hope to show you on a single slide. But to show you things like that we're gating out monocytes from lymphocytes, CD4 from CD8, some other T-cell differentiation markers, and then various combinations of cytokines are shown here for CD4s, for CD8s. And you can see that in this particular individual, there's a pretty nice CD4 response to CMV. The cells are making interferon gamma, some of them IL-2, they're making TNF, and much smaller subsets making GM-CSF. And in the CD8s, the response is smaller overall, but has a similar kind of pattern of cytokine expression. But obviously, this is just the beginning of the analysis, and we are using tools like SPADE and other kinds of clustering algorithms to get a better handle on this data and ask essentially what are the main phenotypes of responding T cells in a particular sample or across a particular group of samples. Now we're also developing the ability to look at PhosphoFlow by CyTOF, which is something that the Nolan Lab has done a lot of. And standardizing this assay has not been that easy for us to be able to look at all of the different cell surface markers that we want to be able to look at in combination with the signaling markers. And then in particular doing that in whole blood. So this is kind of our latest effort, which is led by Rosemary Fernandez in the lab, and which is a whole blood phenotyping plus cell signaling panel. And it identifies, as you can see here, essentially 32 different cell subsets within whole blood, from the different types of granulocytes to various B cell subsets, T cell subsets, Tregs, dendritic cell subsets, and NK subsets. So a fairly diverse, not very deep dive, but a fairly diverse categorization of immune cells. And then for any one of those particular gated populations, we can read out a matrix of different stimuli and different phosphorylation nodes, which we've now expanded beyond the cytokine stimuli. We're doing things like CD3, CD28, and LPS to look at some of the TLR signaling pathways. And in addition to the stats that we have in the conventional panel, conventional fluorescence panel, we now have also some other phospho readouts, ERK and phospho-S6 and some others. So it becomes a rather broad fingerprint of the immune response of that particular person's blood. And we think that this is going to be a highly useful assay in any number of different diseases because it is going to be such a broad yet detailed look at the signaling in whole blood. All right, so moving on to the protein level, our major technique, as I mentioned here, is Luminex. And with the help of Yael Rosenberg-Hassan, who's our technical director and manages the immunoassay group, we're now moving to a 63-plex human cytokine array. For those of you who aren't familiar with this platform, the way it works is that you have these hard-dyed beads which can be colored. They're impregnated with dyes, 2 different colored dyes in combinations, such that each individual bead in this array has a different specific color address or different combination level of these 2 different dyes that allows the software to identify it as having then a particular marker or a capture antibody associated with it. So then each bead becomes its own little immunoassay with a capture antibody capturing a cytokine or other marker, and then a biotinylated detector antibody with a fluorescent streptavidin bound to it. And that fluorescence is then read out in a little dedicated flow cytometer. 2 of the PMTs are dedicated to reading the bead address, the 2 colors of the beads, and the 3rd color reads out the bound detector antibody. And so in that way, you can multiplex, actually as shown here, up to 100 different bead types and therefore different analytes. But we're using, as I said, now up to 63. So one of the ways that we've used this assay is in a study. This actually was led by Gary Fathman's group of healthy aging. We have here about 200 healthy donors, all aged 40 and above, going all the way up to the 90s. And what you can see when you cluster them, these are all the different cytokines here, and these are the individuals. And when you do basically 2-dimensional clustering, which I think didn't work all that well here because it didn't put all the high-expressing cytokines together, but nevertheless, you can see that there are these groups of highly expressed cytokines and groups of very low expressed cytokines, and then some that are in between. And you also see some heterogeneity. So you see red and yellow spots in amongst the green and blue. And I would point out in particular that there are some patterns here where, for example, these individuals that I've circled now have at least 1, 2, 3 cytokines that are kind of elevated in concert, okay? Unlike their neighbors, which are low for those 3 or 4 cytokines. So these kind of patterns are something that Jake Glanville is helping us to work up and kind of elucidate. They seem to be a general phenomenon in the population, and so he's calling them immunotypes. And it's a way of kind of looking at the population and defining normal ranges in a little bit different way. Rather than saying there's one normal range for everybody, we can now sort of look at these patterns and say, well, if you have this certain immunotype, then this is your normal, but that's a different normal from perhaps another person or another group. Okay, moving on to the gene level. As I mentioned, we do conventional microarrays now, both 1 and 2 color platforms, depending on the application. Human and mouse, we do miRNAs, and we have even a immune-specific array. But these are kind of, if you will, old hat technologies. The thing that we like to do most in the genomics area these days is the Fluidigm Biomark assay, which you may be familiar with. It's a nanofluidic platform developed by Steve Quake's company, Fluidigm, where you basically put a series of samples in these inlets on one side of the chip, and then a series of qPCR reactions on the other side. And the instrument makes all of the microfluidic connections between these to give you this big grid then of samples times genes that you're looking at. And you basically get, for very minimal pipetting and very minimal input reagent, a large amount of data in terms of qPCRs. You can do this because the volumes are so small. You can do this at the single cell level. And so we have several projects now which are looking at, for example, sorted tetramer positive T cells in specific, for specific pathogens or in allergy, and then essentially looking at key genes that would phenotype those particular T cells and look at their heterogeneity or the commonalities between them. And this is an example of some such data that we're doing in collaboration with Kari Nadeau's group in food allergy and peanut allergy, where this is an example of one person at baseline before they begin immunotherapy, and here 3 months after immunotherapy. Each line is a sorted tetramer-positive T cell, and I haven't labeled all the genes across the columns here, but I'm pointing out 2 genes of interest, FOXP3 and IL-4, and I don't know how well you can see it from the back, but at baseline, this particular patient has a fair number of IL-4 producing cells, only 1 out of this group producing FOXP3, and after 3 months of immunotherapy, that seems to be shifting somewhat, where there's no more IL-4 producers, but there's now a few more of the FOXP3s. So we're going to look for whether these kinds of patterns are generalizable. I don't think it's as simple as just this. When we have more patients, we're seeing more heterogeneity of patterns, but we obviously can try to pick apart the commonalities. And what we're doing now, as I alluded to in the beginning, in combination, we're doing T-cell receptor repertoire analysis. And this was developed by Arnold Hahn in Mark Davis's lab, along with Jake Glanville. And so for each of these individual cells, you have a corresponding TCR sequence, and you can start to associate phenotype with specificity, and you can look at persistence of clones over time and things like that. So we think that's going to be an interesting direction. All right. I'm going to spend the last few minutes talking about the data analysis challenge, because as I said, that was our third aim, and that's one of the biggest roadblocks that we have when we collect these very comprehensive data sets. And so the main tool that we've developed in collaboration with Janet Siebert, who has a company called Cytoanalytics, is this database that we call Stanford Data Miner. Basically, it has an interface that uses an open source package called OLAP. It stands for Online Analytical Processing. And it's a way of displaying your data and rolling it up in a fashion in these sort of interactive tables. And the idea is that you can choose the dimensions that you want to show in both rows and columns, and you can filter the data on particular attributes or projects, for example, as well as pick different readouts in terms of statistics on the data you're looking at. So here is data from a particular study. It's actually this Fathman aging project that I showed the heat map on earlier. And it's a very high-level view in which we've segregated out the data by gender, and you can see the number of data points for each of 3 different assays. But this data is not very meaningful beyond a kind of event chart view. What you can do, though, is you can click on the plus sign here and then expand all of the individual analytes under any one of these assays, and then see the rolled up mean of the data for each of those assays. So here, which I know you can't read, is the beginnings, at least, of the list of analytes for the CyTOF phenotyping assay. It actually would stretch down to almost the floor, but I couldn't fit it all. And it's divided into B-cell analytes, other, and T-cell analytes. And so for each of these now, we've actually expanded the display of statistics to include things like the min and the max, the number of outliers, so that you could look and see, oh, you know, there's 12 outliers for this particular B-cell subset, and there's 13 outliers here. I wonder if those are some of the same people. And so what you can also do then with these rolled up data is you can click on any individual cell and see the underlying data points. So you can get down to the level of individual data points as well as having rolled up or aggregated data. And all of this is, of course, downloadable as a kind of Excel spreadsheet. So you can then take it into other kinds of analyses. And we also have now some other visualization tools, because it's one thing to look at a big list of numbers, but it's nice to be able to visualize the data. So that very same aging study data, if we take just the Luminex data out of there and display it in this view that Janet has, which is called parallel coordinates, and we break out now, again, the gender, the male and female. And what the parallel coordinates view that Janet implemented here does is it picks for you the top analytes by CV. So these are the analytes that vary the most within your data set. Hence, they're probably the most interesting ones to start looking at, okay? And what is particularly interesting in this data set, when you break it out by male and female, is that, first of all, you see there's a lot of data down here where many of these cytokines are very low, but then you have these high outliers. So especially for leptin and for PDGF-beta-beta, there's a subset of people that are really high, okay? Secondly, again, among these top 10 analytes by CV, the high outliers are almost exclusively females, okay? The exception is PDGF-beta-beta, where it's higher in general in males, and the high outliers tend to be male. But in the other cases, they're all females. So this is something that you didn't see in the heat map I showed you, because the scaling of the heat map truncates these, and you don't see these half dozen or 10 outliers amongst a group of 200. They show up no higher than some of the ones that are down in here. But when you do this kind of a view, you see them. And furthermore, what you can do with this kind of view is you can select a subset. So let's take the 4 highest ones for one of these analytes, interferon beta, and you see where they lie on the other coordinates. And what's interesting is that at least many of these are what I would call serial outliers. They're not just high for interferon beta. They're high for maybe MIP1 beta, maybe IL-17F, maybe leptin. So there you can start to discern patterns, again, getting back to this concept of immunotypes, that there may be certain cytokines that are traveling in parallel, and that people who are high outliers for one may be high outliers for a couple of others. So I think this is a good visualization to help you start to appreciate that. The other tool that Janet has built into this database is a decision tree algorithm. And this is, for those of you not familiar, a machine learning tool that allows you to use your data set and try to predict a particular outcome of interest. And in this kind of toy example here, we're asking, using a set of cell phenotyping data, whether we can predict age, okay, in 2 major buckets, under and over 40. So can the data tell us which subjects are over and which are under 40 based on their cell phenotyping? And as it turns out in this data set at least, these 3 different CD8 T cell subsets, using the breakpoints that are shown here, do a pretty good job of categorizing people into the subsets under 40 and over 40. And this is then something, you know, it's not definitive. You would have to go into other data sets to see whether the same predictors hold true, and whether this tells you something about biology. But in this case, it's actually not too surprising, because these are subsets that are known to change with age. Naive cells declining, and these CD27 positive cells disappearing with age, for example. So, so that's another kind of nice tool to have when you have created a set of data, if you will, selected in the OLAP view, filtered on particular things, and then take that data set and ask what you can predict with it in terms of a machine learning tool. All right, so I'm going to stop there and put us a little bit ahead of time. I should mention that the schedule may have been a bit intimidating, because it was pointed out to me that there was no break. We will have a break after the 3rd talk, and so hopefully I have gotten us a little bit ahead of schedule so that we'll have time for that. And I just want to finish by acknowledging the folks in the HIMC. We have a quite big group now, and in particular point out Mike Leipold, our CyTOF manager, Rosemary, who runs the phospho-CyTOF and phosphoflow assay in particular, and Yael for the Luminex and other immunoassays, and I think that's the main people whose data I showed. The collaborations, which I've mentioned along the way, and in particular our faculty advisor, Mark Davis, and the database development by Janet and Wes. So thanks for your attention. I'm happy to take a few questions now before we move on to Yael's talk. Are there any questions? Yes?
So you mentioned you had some issues
Well, the main one really was being able to get good staining for the cell surface markers in the context of fixed epitopes, right? Because you do this 15-minute stimulation and then you fix, and now there's a lot of markers that don't look so good after fixation. Things like CD4 and CD8 are fine, but there's, a lot of the CCR7 and CD27, some of these other markers that were a little bit hard to work out. So we ended up, going through multiple clones to find the best clone, and then titering it very carefully, and also, the number of washes to try to keep the background low. So really getting those things optimized was pretty important to get good staining. The phosphos actually worked pretty well with methanol perm.